Hospital. Yeah. So today, my topic is going to be immunoglobulin classes and class switching. So to begin, we have to know about the structure of the immunoglobulin. So every immunoglobulin consists of mainly two chains. One is light chain and one is heavy chain. And the light chain of the immunoglobulin consists of one variable region and one constant region, whereas the heavy chain of the immunoglobulin consists of one variable region and three constant regions. Between these light chain and heavy chains, there is a disulfide bond. There is a antigen binding or FAB region, which acts as the binding site for the antigen, and the other region is known as the FC region, which expresses for the biological activity of the antibody molecule. So this is the variable region of the light chain, and along with the variable region of the heavy chains consists of the paratope, which binds the antigen to the antibody molecule. Now moving on, there are few classes of antibodies that we know, and these classes are determined mainly by the presence of the nature of the heavy chain in them. So each of the antibodies contains different variation of the heavy chains in them, and as according to the presence of the nature of the heavy chain, the antibody molecule is determined. So coming to the first class, which is known as the immunoglobulin G, is the most abundant immunoglobulin and consists of around 80% of the serum immunoglobulin's volume. There are many subclasses of the IgG molecule as well, like 1, 2, 3, and 4. But mostly IgG1 and IgG3 along with IgG4 can cross the placenta, and IgG is only of the antibody which can cross the placenta. IgG3 is most effective, the complement activator, and IgG1 and IgG3 has high affinity for the FC receptor on the phagocytic cells, and also they are good for opsonization. Coming to the next class of antibodies, which is known as IgM, they consist of around the 5 to 10% of the immunoglobulin volumes, and it is also expressed on the surface of the B cells. It is the primary antibody to appear in case of an immune response, and they are usually pentameric or hexameric, and they are having the most heavy molecular weight among all antibodies. It is often found in the blood in a pentameric structure, which increases the surface area and the number of sites for antigen binding. IgM is therefore particularly good at trapping neutralizing antigen and also activating complement, but is less good at ADCC and opsonization. Coming to the next antibody, which is IgA, it is consisting of the 10 to 15% of the serum immunoglobulins, and it is predominantly found in the surface mucosa, milk, saliva, and tears. It is mainly dimeric in nature, and it can be also monomeric as well. Now, this picture shows that IgA transport across the cell. So I'm not going to delve into detail of that because it will consume some time. Now, the next class of antibody is IgE, which is very low in serum, participate in immediate hypersensitive reactions like asthma, anaphylactic shocks, and etc. It mainly binds mast cells and blood basophils through the FCE receptor. Binding causes degranulation of the mast cell, and ultimately that gives rise to histamine release. Another type of antibody is IgD, which is expressed along with IgM on the beta cell surface. And we do not know any other biological effector activity, and it has a very low level of serum concentration as well. So this is how IgE can act. It binds with an allergen, and the FC receptor specific for IgE binds with the mast cell, and then it actually degranulates the mast cell and effectively releases histamine from the mast cell and usually aggravates the allergic response. Now, antibodies can act also as immunogens. They are like isotypic, allotypic, and idiotypic. Isotypic is when if you inject an antibody in a different species, an anti-isotype is generated. Allotypic is when an antibody is injected in the same isotype within one species of small differences. And idiotype is when an injected monocular antibody into a genetically identical recipient, then the anti-idiotype antibodies are generated. Now, coming to the most important topic of today, which is class switching. So we have to know that the class switching, the way of the class switching occurs is also known as isotype switching, or isotype combination mechanism that changes a B cell's production of immunoglobulin from one type to another, such as from the isotype of IgM and the isotype of IgG. During the process, the constant region portion of the antibody heavy chain is changed, but the variable region of the heavy chain stays the same. This mutation or recombination is known as the VDJ recombination. And it's an irreversible process, means that once antibody is class switch, it cannot get back to the previous class. The mature naive B cell expresses the IgM and IgD on their surface. On primary response, responding to antigen and T helper cells, some activated immunoglobulin secrete IgM, the dominating antibody in primary response. I can see just it's obstructing my view. Can you change this? The secondary response is dominated by IgG, IgE, and IgA secreting cells. The class switch recombination eliminates the other C regions of the heavy chain, and retains only the right class. It does not affect the coding sequence of variable heavy chain region. It allows more types of antibodies to be produced with the same antigen recognition as well. Now, this is the way how class switching occurs. It actually cleaves at the switch points, and the remaining portion is joined by some enzymes and expressed in a totally different form. Usually IgM is secreted at first, and then it is switched to any other kind of a molecule, such as IgA, which has been shown in this picture. This is the same picture presented in a different format. So this is a transcript for IgM, and with some enzyme, the switch sites are cleaved, and the remaining portion of the molecule is rejoined and codes for another kind of an antibody, which is IgG in this picture. Now, this is a mechanism in short. Portion of the antibody heavy chain locus are removed from the chromosome. Double-stranded breaks are generated in DNA. DNA is nicked and broken at two selected S regions by the activity of series of enzymes. Intervening DNA between the S regions is subsequently deleted from the chromosome, and free ends of the DNA are rejoined by the process called non-homologous end joining. Now, there are various cytokines that are responsible for this class switching actions, such as IL-4, IL-5, TGF-beta. These are main interleukins that mainly takes part in this class switching process. TGF-beta mainly gives rise to the growth of immunoglobulin A, whereas interleukin-4, interleukin-13, and interleukin-1 stimulates the growth of immunoglobulin E type. So that's all about class switching. So in addition to the highly repetitive structure of the target S region, the process of class switching needs S regions to be first transcribed and then spliced out of the immunoglobulin heavy chain transcripts. Chromatin remodeling, accessibility to transcription, and to aid in the synapses of broken S region are under control of the large super enhancer located downstream to the more distal C alpha gene at the 3' region. Class switching is a process which takes place in B cells.
8 minutes.
Yeah, to alter the class of antibody produced during an immune response from IgM to any other class. This is pivotal process that allows the response to become tailored to the infection that is present. Each class of antibody therefore has its own strengths and function, including opsonization, ADCC, and neutralization. The choice of antibodies directed by the infecting pathogen, as well as the signals from surrounding immune cells, and occurs via an irreversible process called as class switch recombination. Thank you.
Okay.